আমি তো দেখতে পাচ্ছি না, তোমরা বাকিরা কেউ দেখতে পাচ্ছ?
না না না। আর স্যার কন্ট্রোলের অপশনটা কেন আসছে?
কন্ট্রোল কিছু করো না,
কিছুই করো না তোমরা, ওটা ওই টেক কন্ট্রোল অপশনটা তুমি
ঝামেলায়
ইয়ে
পড়ে
করে
যাবো।
দাও, অফ
না
করে
না,
দাও।
ওটা তো থাকে না, আপনি
Take
স্ক্রিন
control
শেয়ার করুন।
হ্যাঁ, আমি তো কিছু দেখতে পাচ্ছি না এই সমস্যা, আমার আবার নেটওয়ার্কের কোনো ইস্যু হচ্ছে না।
এবার দেখো তো একটু, ম্যাডাম একবার দেখবেন।
হ্যাঁ, আমি তো দেখছি কিন্তু দেখতে তো পাচ্ছি না কিছু।
আসেনি স্যার, আসলে বলবো। এসছে এসছে।
হ্যাঁ, এবার এসেছে।
হ্যাঁ স্যার,
হ্যাঁ, এবার এসেছে।
ঠিকঠাক।
School of Health Sciences offer. এটা এসেছে। যেটা দেখতে পাচ্ছি আমি তোমায় স্ক্রিনশট নিয়ে পাঠিয়েছি, দেখো এটাই। আর কিছু শো করতে চাইছো না? এটাই ঠিক আছে?
হ্যাঁ ম্যাডাম, আমি দেখলাম না, আমি এর ফুল স্ক্রিন মোডটা শো করতে চাইছি, আমি তাই ফুল স্ক্রিনটাই শেয়ার করি। আমার কথা বা শোনা যাচ্ছে?
Yes, please. Yes.
I think it is now okay.
হ্যাঁ, এটা ঠিকঠাক আছে একদম।
Okay. So let's wait for some time. I think within the time our guest will join.
I guess he had joined. জয়েন করিতে ছিল। উনি অনেকক্ষণই আমি 3:16 জয়েন করেছি, উনি তখন থেকেই জয়েন দেখাচ্ছিলেন। I don't know কি আবার বেরিয়ে গেছেন কিনা somehow. উনি ছিলেন জাস্ট বোধহয় এই মুহূর্তে, না না উনি আছেন, তবে ওনাকে তবে উনি অ্যাটেন্ডই করা আছেন। প্রণব, ওনাকে প্রেজেন্টার করতে হবে।
Hello, Mr. Sarat. Am I audible?
Yeah, you are audible.
Hi. Hello, everyone.
Hello, sir.
Yeah, you're audible. Am I audible? Do you think I'll need to connect an external audio phone? Is this all right?
My voice is clear or not? Please, anyone respond.
Your voice is clear.
Yes, clear. Hello? Yes, Honorable sir. It's okay. Fine. Hello? Am I audible? Yes, you are audible. Thank you. Okay. So, good afternoon, everyone. Grand welcome, Mr. Sarat. Today, we will be conducting our third corporate lecture series. And before we start, I would like to introduce him to all of you. Mr. Sarat Raghunathan is currently a molecular technologist, All Nile Medical Complex in Oman, and he would today deliver a lecture on molecular diagnostic method for COVID-19 detection. I guess it's a very, very relevant topic in today's time. So he is a research scientist with a bachelor degree in the field of genetic engineering and he earned that degree from SRM Institute of Science and Technology. His area of interest includes antimicrobial resistance, medical microbiology, molecular microbiology, and synthetic biology. He has a huge experience in various labs all over India regarding the field of microbiology and advanced molecular biology. He had also worked with Care Reference Laboratory in Kerala as a senior technology specialist and also in King Institute of Preventive Medicine as a laboratory technician. He is currently focused on RT-qPCR for COVID-19 samples and its reporting for continuous samples. So we hope we would all enjoy his lecture, and students and all other guests who are present here, we hope to have a very wonderful interactive session. And also students are encouraged to ask questions, okay, after the lecture is, once the lecture is over. So you start. And so welcome, Mr. Raghunathan.
Yes, thank you so much for the introduction. Do you think I'll take over? Or, hello?
Mr. Raghunathan, I should welcome you in the university. Although we are conducting, these things, virtually. Let me introduce myself first. My name is Professor Niloy Sarkar. I'm the Dean Academics of this university. So I would like to welcome you on behalf of entire fraternity, right, on this virtual mode. And I'm very sure, through your lecture, the student will get that insight, and they'll, apply this particular knowledge in their working field, and they'll be immensely benefited out of it. And in case, we'll be expecting your, physical presence, inside the campus. So I got this intimation that you are in Oman now. So whenever you'll be visiting India, so please try to come. I believe, by that time, we'll be opening, our university physically also.
Pleasure.
So thank you very much, for your, coming and for your agreeing to this aspect to give a lecture on molecular biology. I wish you all the best. It is over to you. Thank you.
Swarnabha here. I have made you the presenter of this meeting, and also you can share your screen now. Okay.
I'll do that now. Yeah. I believe everybody can see the screen. Is it visible? Yeah. Is it all correct now? My voice is clear?
Right.
Yeah. Yes, sir.
Yes. Yes. All right. Great. Hello, everyone. I'm Sharath Raghunathan, and I believe by now you guys understand who I am. And thanks. That was really nice of you to introduce me. And I'm really happy to be a part of this. And I'd like to thank Dr. Biswas for inviting me for this lecture series. I'm really honored. So today's topic is molecular diagnostics of COVID-19. So as you all know, we are in the middle of a pandemic, and we already faced 3 waves, and we are on the 4th wave. So I thought if I can explain you about the nuances and how molecular diagnostics of COVID-19 is done. It's basically done by you guys. Like maybe after 2 to 3 years, it's maybe basically you guys are going to do this. So I'd like to explain you what are all the basic nuances of COVID-19 and applications and the future prospects of molecular diagnostics. Maybe not only COVID-19, for other diseases. So that's what I'll be discussing in today. I have actually split my presentation into 2 different parts. I'll be introducing with this slide, and then we'll move on to a different presentation, and then we'll be back to the same. So I'll just move on. So the first topic I'll be explaining you these topics today. So I'll be explaining you about the COVID-19 diagnostics first, and then we'll move on to what is molecular diagnostics, what are all the basic techniques required to successfully continue with a molecular diagnostic procedure. And then with regarding to COVID-19, what are all the genes involved in COVID-19 diagnostics? You know, there are a lot of genes inside the virus, but then we cannot use all of them to efficiently diagnose the infection. So we use a specific set of genes. So I'll be explaining you how we came to that particular genes only, and then what are all the roles of that particular gene. So we'll be talking about that today. And then other than RT-PCR, what are all the other futuristic techniques that are used for molecular diagnostics, not only for COVID-19, but then can be used in a wide range of other aspects. So, and then I'll also be introducing you to a few job opportunities for you in the future. So this is what we'll be talking about today. I hope we'll have an interactive session. So about COVID-19 diagnostics. So as you can see on the top, approved diagnostic methods. We have only these 3 diagnostic methods that are approved by the WHO, FDA, and in India, ICMR approved techniques are rapid antigen testing and RT-PCR based molecular testing, and then immune enzymatic serological testing. That's basically like our ELISA. So we have these 3 testing that is approved, and you will need the result of at least one of these tests to for the doctor to give you a standard advice. Otherwise, if you give some other test, the doctor will not have a standard protocol to give you a prescription. So these are the 3 tests in which the infection range, the amount of viral load, all those things are standardized. So first one would be rapid antigen testing. Then we have RT-PCR. Then we have ELISA. Then other than these 3, we also have a few other methods like viral culture, next generation sequencing methods, radiological investigation, and clinical examination. But then these are not currently in use because these were firstly used as these are the initial methods of identifying an unknown strain. If you need to know what kind of infection is going on, what is actually happening, to know that, you will have to first identify what is the organism, whether it's a bacteria or it's a virus. So for that, you'll have to undergo culture methods. After undergoing culture methods, you'll go for like microscopy, you'll understand what organism it is. You'll go for other biochemical assays, and then we'll go for next generation sequencing. Next generation sequencing will let us know whether we have encountered with this virus before or not. What is the particular genome of the virus?